This method facilitates the effective characterization of disease-related RAF kinase mutants. This technique is highly sensitive and reproducible, cost-effective, user-friendly, and does not require any advanced equipment.
To measure the catalytic activity of RAF mutants, introduce flag tag mutations into the RAF coding sequence by PCR before inserting whole sequences into the appropriate vector according to standard protocols. Insert the glutathione S transferase coding sequence upstream of the RAF mutant coding sequences to generate vectors encoding glutathione S transferase fused RAF mutants in the same manner. Transfect 80 to 90% confluent 293T cell cultures with the vectors encoding the flag-tagged RAF kinases or their mutants according to standard protocols. After 24 hours of culture, replace the medium in each culture. After 48 hours of culture, add 400 microliters of lysis buffer supplemented with protease and phosphatase inhibitors to each well on ice. After 5 to 10 minutes, transfer the cell lysates to a 1.5 milliliter tube for centrifugation to sediment the cell debris. Transfer 300 microliters of each sample into individual 1.5 milliliter microcentrifuge tubes. Set aside 40 microliters per sample for downstream whole cell lysate phosphoextracellular signal-regulated kinase 1 and 2 expression and activity immunoblot analysis. Add 20 microliters of anti-flag affinity beads to each tube for a 1-hour incubation with rotation in a 4 degrees Celsius cold room.